Good afternoon, ladies and gentlemen. Thank you for joining us today for this webinar session on the interesting topic, long-term drivers of above-ground, below-ground linkages and ecosystem functioning. This event is brought to you by the NTU Institute of Advanced Studies, Asian School of Environment and Earth Observatory of Singapore Research Fellow Club, and the Graduate Students Clubs of the School of Physical and Mathematical Sciences and the School of Biological Sciences. My name is Shivam and I'll be your host for this webinar. Before we begin, there are some housekeeping rules for this webinar. All of your mics have been muted to prevent any accidental disturbance. If you'd like to ask a question during the Q&A session, please use the raise hand feature at the bottom of your Zoom screen. When your name is called, please ask your question. Or you can also post your questions under the Q&A tab at the bottom of your Zoom screen. For the postgrad students who require the endorsement of the attendance, please scan the feedback form at the end of the session and indicate the need for endorsement. Today's webinar is being organized as a part of the Discovery Science Seminar Initiative. Now let me introduce the speaker for today's webinar, Prof David Wardle. Prof David completed his PhD from University of Calgary in 1989 and is currently a Professor Smithsonian Chair of Forest Ecology at NTU. His research projects focus on the community and ecosystem effects of invasive animal and plants, the ecological consequences of wildfire, ecosystem changes across successional, retrogressive, and elevational gradients, aspects of island ecology, and the ecological role of forest understory vegetation. He has published around 400 research articles in reputed journals. From 2006 onwards, he has always been among the world's 10 most cited researchers in ecology and environmental sciences. He has received many accolades during his career. Recent ones are 2018 Eminent Ecologist Award from the Journal of Ecology and 2020 Whitaker Distinguished Ecologist Award from the Ecological Society of America. Without further ado, let's welcome Prof David. Prof David, please.
Thank you. You can hear me okay?
Yes.
Great. Okay, thanks very much. The presentation that I'm going to give is going to consider the long-term drivers of above-ground, below-ground linkages, drawing on the work that my colleagues and myself have carried out in this area over the past 30 years or so. But before I begin, I'd first of all like to thank the Graduate Student Club and the IAS for the invitation to give this presentation and make it possible. And I'd also like to give a particular call out to Shivam, Chris and Ruchi, who have done all the hard work behind the scenes in organising this event. So in this presentation, I'm going to have two sub-themes meandering your way through it. The first of these is the above ground, below ground approach, the conceptual framework that my colleagues and myself use with regard to this work. It recognises that the plants and animals that we see above ground influence those organisms in the soil, both in terms of those that decompose and break down organic matter and release nutrients for plant uptake, as well as those more tightly associated with plant roots that influence plant growth, such as pathogens, root feeders, and special fungi we call mycorrhizal fungi. I'm going to mention these a few times in my talk, but these are fungi that form a symbiosis with plant roots and help plants take up nutrients to make them and make them grow better. So these organisms interact to influence the plants and animals that we see above ground, and these combined the feedbacks between the two components drive ecosystem processes both above ground, like production, and below ground, such as carbon storage, carbon fluxes, and nutrient cycling. The second sub-theme is that of timescales. Now, the kinds of experiments and observations we do range from a few hours to several years, the lifespan of our experiments, the lifespan of our measurements. So these are processes such as decomposition, nutrient cycling, herbivory, vegetation change. And these both respond to and drive processes that occur over much longer timescales beyond our lifespans. Things such as long-term succession, vegetation processes, and geological and evolutionary processes. So in this context, I'll describe three pieces of work my colleagues and myself have carried out in this area. The first of these is on invasive mammals in New Zealand over timescales of decades to centuries. Then I'll zip across the planet to northern Sweden and describe work we're carrying out there on lake islands and ecosystem processes, centuries to millennia. And then I'll talk about longer term processes still, those which occur over longer term, formed over longer term, such as geological timescales, such as soil aging and climatic gradients. So firstly, with regard to the New Zealand situation. Now, New Zealand is very geographically isolated, and as such, it doesn't have large mammals. The only mammals in New Zealand, the entire mammal fauna, consists of 2 species of bats. We did have a third one which went extinct a little over nearly 60 years ago. So, whereas other large land masses have large mammals that feed on vegetation, in New Zealand, large birds evolved to do this role. So these were the giant moa birds. There were 11 species in total, and these were really big. The tallest stood at 2.5 meters. These served as the big herbivores in the New Zealand context. And these were hunted to extinction by the first human settlers in New Zealand about 700 years ago, about 100 years after they arrived. And they supported other biota as well. For example, the giant New Zealand haast eagle, the largest bird of prey to have existed, which fed on a diet largely of moa birds. And it went extinct when the moas went extinct and their habitat got lost. New Zealand then went about 500 years without large herbivores until the European settlers introduced a whole range of new ones. These include goats, introduced as livestock, and now maintain significant wild herds throughout the more fertile forests of New Zealand. Deer, several species were deliberately introduced for sport hunting, and these now maintain wild populations throughout our forests. Australian brush-tailed possums, intentionally brought over to generate some sort of fur industry, and now serve as a major pest through serving as a boreal predator in our forests, and various rat species that I'm going to talk about a bit later on in my presentation. Now, to understand how these animals affect ecosystems, our tool of choice are exclusion plots. These are fenced plots to keep out large animals. For example, this is a fenced plot to keep out Australian brush-tailed possums, which we've had running for about 15 years. When the possums are not present, they've been excluded. We have lots of grassy plants because the possums eat the grasses out. And in the presence of possums, we have shrubs instead. This shows the effects of European red deer. They cannot get here, but they can get here, and they eat out the understory. And this shows vegetation hedging by invasive goats on Mount Taranaki. No goats, goats. So what we have done is gone throughout New Zealand and selected 30 long-term deer exclusion plots. These were fenced plots set up by the former New Zealand Forest Service in the 60s and 70s, 20 by 20 metres, and they were set up to exclude deer so that folk could look at the effect of deer control on vegetation recovery. So we've selected 30 of these exclusion plots throughout New Zealand, representing most of New Zealand's forest types and spanning most of the New Zealand's climatic regions. What we've looked at for each exclusion plot is the effect of deer on the plant community, the consequences of this for litter entering the soil or dead leaves, the consequences for microbes and the soil animals that feed on microbes and each other, and also feedbacks of that aspect of plant performance. Now, this shows the kinds of plants that the deer reduce and promote. These species won't mean anything to most of you unless there's the odd kiwi out there somewhere in the audience. But basically, these are plants which the deer reduce because they like to eat them, they're palatable, and these are large leaf dicots. These are plants with big, palatable, tasty leaves. When the deer are present, they eat out these species and they get replaced by other species because the competition is reduced, small leaf dicots, monocots, and ferns, plants with much tougher leaves that are far less palatable. And these plants also produce dead leaves that break down more slowly and are less palatable for the decomposing microbes that would otherwise break them down. So what are the consequences for the soil system? Well, this above ground interaction influences many groups of organisms below the ground as well. This shows the responses of various groups of soil biota to deer across the 30 locations. And what this shows is the number of locations out of 30 for which the deer have a significant negative effect, no effect, and a positive effect. When we consider things with small body sizes like microbes, nematodes, rotifers, copepods, some cases the deer have negative effects, some positive. So a range of effects are possible. When we consider things with big body sizes like collembola or springtails, mites, gastropods, that's slugs and snails, spiders, certain beetle groups, the effects are consistently negative. We never get these positive effects. This is because different mechanisms regulate small and large-bodied soil organisms. When we consider small-bodied soil organisms, there's a whole range of effects possible. The deer can have effects which affect resource quality entering the soil, quantity, successional pathway. These effects can be positive or negative. Different mechanisms dominate in different environments. However, the invasion by deer has only negative effects on the big-bodied soil organisms. And this is because deer have a strong physical effect by treading, squashing things, destroying habitats and such like. And these effects are only negative. This leads to an interesting comparison between what the invasive mammals do and what the extinct moa birds used to do. When we consider the moas, they were really big. They were bigger even than a deer, so really big birds, but they have large feet that spread out over the ground surface. So their foot pressure is a lot less. The deer both have high foot pressure and they also have really high densities owing to their predators, owing to them not having much predation. So that means that the deer, large-bodied soil invertebrates are being subjected to a form of disturbance that did not exist prehistorically. For some reason, I can't change the next page. Yeah, I think my screen seems to have frozen. Oh, no, that's it. Great. Okay. So this in turn has important feedbacks for plant growth. Now, this summarizes some work that's essentially headed up by my then postdoc Paul Cardall. But what we looked at, or what he looked at, was the effects of deer on plants manifested through the soil. So the deer have a direct nutrient input effect, but they also have strong soil compaction effects through their hooves, squashing soil, destroying the habitat. Now, what his work shows is that this negatively affects soil physical and chemical properties, negatively affects soil biota, and this has negative effects on plant growth because there's less nutrients being cycled, less nutrients for the plant to uptake, and plant performance is therefore impaired. Further, he showed that the mycorrhizal fungi, the special fungi that form a symbiosis with plant roots, were impaired, and this in turn impaired plant growth. So the tool we use, exclusion plots, can be used not just for looking at invasive animals, but also for overabundant animals. I'll just briefly flag some work we're currently in the process of setting up actually in Singapore. And this involves wild boar. As I'm sure most of you are familiar, if you walk through our local parks, there's enormous numbers of wild boar in the forests. And these have high densities in part because their predators are no longer present. There's no longer tigers walking around eating them up. Now, when boar are present, they have a strong effect in the forest because they cause a lot of ground disturbance, cultivating, looking for grubs and roots and such like. And they also affect soil respiration and soil carbon. So this shows a study, it's not from our work, it's actually from China. But what this shows is soil respiration, carbon dioxide released from the soil when there's no wild boar here and when there is wild boar. So the digging of the ground by boar causes a lot of carbon to be lost from the soil, so there's less carbon sequestered in the soil. Sequestration of carbon in the soil is, of course, a good thing because this helps mitigate atmospheric CO2. what we're in the process of doing is building long-term exclusion plots throughout Singapore. These are fenced plots that we've having constructed, which reach about 110 square meters. Over the past 3 months, we've had 20 of them set up in the central catchment area in Selangor, McRitchie, Bukatima, and we're in the process of having another 10 constructed on Pulau Ubin. 5 have already been constructed and the other 5 are being built as we speak. 5 of them on the eastern part of the island that we can all get to, and 5 on the other half that's closed off for the general public for conservation purposes. So we don't have any data yet, but what we're hoping to over the next few years is understand what effects these wild boars are having on above ground, below ground processes through their intensive cultivation of soils. Okay, and now move on to another invasive animal story. And this is the so-called Rats and Seabird project, a project that we've been running off islands on the northern coast of New Zealand now for the past 15 years or so. Now, on these islands, like many coastal systems worldwide, the main ecosystem driver is seabirds. And these seabirds have 2 main effects. Firstly, in the daytime, they forage in the ocean for fish, and at nights they come and settle on the land and deposit guano as fecal material, and therefore add carbon and nutrients to the ground. The second effect is that during the nesting period, they make a dense labyrinth of burrows in the ground. These are all forested islands, but they go into the forest, to the forest floor, they cultivate the soil and make these burrows where they have their eggs and where they lay their eggs and raise their chicks. So they cultivate the soil a bit like a giant earthworm would. Now, some of these islands still have really high densities of seabirds, and others of our islands, like many coastal systems worldwide, have been invaded by rat species. So we've got 3 rat species. The first is the Pacific rat, introduced by the first human settlers about 800 years ago. And the 2 species that are a lot more competitive and have competed them out, the Norway rat and the ship rat that have been introduced by the European settlers. When these rats are present, they have really negative effects against the bird life because the birds have not evolved with them. And so they eat the eggs and the chicks of the seabirds. Consistent with this, we have 9 islands where the rats have never invaded, and we have really high densities of seabirds. We have 9 other islands where the seabirds have invaded. They've eaten the eggs and the chicks of the seabirds, and the seabirds are pretty much gone. This has strong effects on soils. For example, if we look at soil carbon and nitrogen, the soil has a lot more carbon in it, so the rat-free islands, than the invaded islands, because seabirds are no longer transporting carbon from the ocean to the land. Same with regard to nitrogen, a lot less on the rat-free, a lot more on the rat-free islands. This is also a difference in terms of stable isotope. The stable isotope signature of the soil is different in the rat-free and the rat-invaded islands because marine-derived and terrestrial-derived nitrogen have different isotopic signatures. Now, these effects manifest themselves through the soil food web. So we've got an above-ground interaction, the rats eat the seabirds. This also affects food webs in the soil. This shows work by my then postdoc Tad Fukami, which looked at the effects of rat predation on seabirds on multiple trophic levels of the soil food web, different types of soil organisms. This diagram is not as complex as it looks. Basically, the blue bars are the 9 seabird islands, the red are the 9 invaded islands. So when the above ground system is invaded, this cascades through the soil food web, and we get reduction in soil biota in many trophic levels. Now, these soil organisms are really important in cycling nutrients, which then fuel plant growth. And consistent with this, we find plants on the invaded islands are a lot less productive. This can be demonstrated by simply collecting soil from each island, taking it back to a glasshouse and growing plants in it. And we find that when plants are grown in rat-invaded soil, they grow less well, and take up less nutrients like phosphorus than they do on the Seabird Island, owing to less nutrients in the soil, but also owing to less microbial activity and nutrient cycling carried out by the soil biota. Now, I mentioned that the seabirds have two main effects. They have a fertilization effect, which is positive for plants, but they also have a disturbance effect, which is negative. They cultivate through the soil, smash up the plant roots, and therefore the trees fall over. When the rats invaded, these effects are mitigated. So consistent with this, we find that the biomass of trees, and we use basal area as a surrogate of biomass, is less on the Seabird Islands than the invaded islands. This is because here we have seabirds cultivating the soil, knocking trees over, reducing biomass. So they're more productive, but less biomass than when the rats invade and reduce the seabird effect. Now, these results are relevant to understanding carbon storage issues. There's been quite some interest in how invasive organisms affect ecosystem carbon storage, because carbon storage is obviously good for mitigating climate change and CO2 enrichment in the atmosphere. And it has been proposed in quite some grey literature that invasive organisms are bad for carbon storage, so controlling them should therefore promote carbon storage. There are, however, few empirical tests of this, but we can test this on our islands, which we believe are very typical to many coastal systems worldwide, dominated by seabirds. What this shows is carbon storage on the islands above ground, and above ground we have more carbon on the invaded islands. This is because the seabirds smash the plant roots up, so there's less standing biomass. Below ground, we have more carbon storage in the Seabird Islands, owing to the seabirds transporting carbon from the ocean to the land and stabilizing it in the soil. When you sum these, however, you have 37% more carbon stored in the invaded islands. Now, this is kind of a perverse result because it means that if we wanted to conserve systems for their carbon storage, then this is actually the state we want, the invaded state. This shows the trade-off between carbon storage and biodiversity, because if we're protecting things for their unique biodiversity, obviously we want these islands. They have the unique seabird colonies, and they also have other unique biota as well, biota found nowhere else. For example, they serve as habitats for the New Zealand Tuatara, of which we have 3 species. These are reptiles that date back to the Cretaceous and have no living relatives on Earth. They are now endemic to a small number of seabird-dominated islands where they live in the seabird burrows. They've long been eliminated from the New Zealand mainland and on rat-invaded islands because rats eliminate them whenever they're present. Another unique organism as well, the giant New Zealand weta. These are large grasshopper-like creatures which are endemic to New Zealand, and they evolved to perform the role that small mammals like mice perform in other systems. This is a species that's among the world's largest insect, and it's now been wiped out everywhere on New Zealand except for 3 of our seabird islands. So this is now endemic to 3 of our islands that we work on. Quite an amazing creature. It's about the size of your hand. So, okay, I'll leave it here for the invasive organism story. And now, staying with the island theme, I'll continue by talking about work we're carrying out on islands in Sweden, again looking at ecosystem processes relevant to carbon storage. This is work we've been carrying out in the boreal zone of New Zealand, of Sweden, for the past 26 years or so. By way of context, the main disturbance in the boreal zone is fire. These fires occur naturally every 50 to 100 years through lightning strike. Some parts of the boreal fires are increasing owing to climate warming and prolonged drought periods. And other places in the boreal, fires are decreasing because people live there, manage forests there, and therefore deliberately suppress fires. Now, the boreal forest stores a lot of carbon, more carbon actually than either the tropical or temperate forests. But unlike the temperate and tropical forest, where the carbon is stored mostly above ground, in the boreal forest, most of the carbon is stored in the soil as humus, as long-term organic matter in the soil. Now, when fires occur, the fires burn off this organic matter, So changes in fire frequency caused by people influence the extent to which the boreal forest is acting as a source versus sink of carbon. Our study system is a group of islands in northern Sweden. These are islands that were all formed by the retreat of land ice about 9,000 years ago. Hold on, just shut the door. Sorry, kids arriving home from school. So just outside the door. These islands are all about the same age, having been formed by the retreat of land ice about 9,000 years ago. And they're all driven by the same extrinsic factors other than fire. So basically we have big islands that are a big target and they get hit off often by lightning. Some of them have burned in the last 50 years, and they are therefore dominated by early successional plants such as pine and vaccinium, plants that grow really fast and allocate their carbon to growth. Now, these are plants that produce dead leaves that are highly favorable for microbial breakdown and break down rapidly. Small islands are a much littler target. Some have not burned for the last 5,000 years, so they move into a different vegetation state, dominated by picea or spruce and impetrum or crowberry, plants that grow slowly and allocate their carbon to defense and lead to slow decomposition and are unfavorable for microbial activity. So this represents some of the very first data we collected from the system. What this shows is decomposition of dead leaves, how fast dead leaves rot. So we get dead leaves, put them on bags and place them on the soil surface or bury them in the soil. What this shows is decomposition of dead leaves on the surface and buried. Each dot is a separate island. So on our small islands with slow decomposition and long time since fire, with slow microbial activity and long time since fire, we have retarded decomposition processes, which leads to a buildup of organic matter. Large islands, decomposition is fast because the plant's presence promote microbial activity. This affects carbon storage on the system. What this shows is carbon storage in terms of island size. These are the small islands. We have slow decomposition, so the dead leaves stay in the soil, they form humus, and we have lots of carbon stored, over a metre in depth. Big islands, decomposition is a lot more rapid. We have shallow humus, and therefore much less carbon stored. This is also apparent if you plot out carbon storage in terms of time since last fire, using the carbon-14 dating of charcoal from each island. Over 5,000 years following fire, carbon accumulates at a more or less linear and unabated rate. It doesn't reach any sort of asymptote. Now, this is relevant to global carbon cycling issues. It has been proposed that suppression of fires in the boreal zone in the northern forest could be causing carbon to be sequestered. And this could explain some of the so-called missing carbon. Carbon that's released by the burning of fossil fuels that remains unaccounted for in global carbon budgets. What our results suggest, if extrapolated over the entire northern zone, is even 200 years without fire can explain the subset of the so-called missing carbon. Carbon that's sequestered and is not accounted for in global carbon models. Now, this is what we thought the full story was until a few years ago, when we sort of started looking more actively at root systems in our study system. We'll start by illustrating the results of a modelling exercise headed up by my then former postdoc, Karina Clemenson. What this shows is the results of the modelling exercise on big islands. Basically, this y-axis is depth down the soil profile, and this solid line is carbon down the soil profile. It can be explained by dead leaves, based on what we know about litter production and litter decay dynamics of the system. The dashed line represents carbon down the soil profile that cannot be explained by plant litter and must have come from somewhere else, such as pumped underneath from plant roots. After about 17 years of age, most of the carbon is root derived rather than comes from above. This is even more apparent for small islands with a really deep humus, carbon down the soil profile derived from dead leaves, carbon that's come from somewhere else, root derived. So most of the carbon is actually root derived, except near the top of the soil profile. And we have multiple lines of evidence that this buildup of carbon from the roots is not actually due to dead root material itself, but actually fungi that are associated with the roots. These mycorrhizal fungi I mentioned a couple of times now, the fungi that form association with roots to help them take up nutrients. We have multiple lines of evidence. I'll briefly walk you through them. First, for each of these panels, the y-axis is depth down the soil profile. Solid line is the big islands with the deep humus. Dotted line is the small islands, with the shallow humus. Dotted line is the small islands with the big humus, with the deep humus. R is what we know for plant root material, and M is what we know for mycorrhizal fungal hyphae. So as we go down the soil profile, the delta C13 signature of the soil, the stable isotope signature of the soil, gets closer to that of fungal material and not root material. The same with the stable isotope of nitrogen, closer to that of fungal material than root material. This is true also with the carbon to nitrogen ratio and the substrate in which the plants are growing. This is close to that of fungal material, particularly for the small islands with the really deep humus layers. We get further insights by looking at eugosterol, which is a material produced by fungi. If we look at free eugosterol, this is eugosterol that breaks down really fast. Lots of into the big islands with the shallow humus, not so much on the small islands with the deep humus. Bound eugosterol, this is eugosterol tied up in really tough, complex forms that's really hard to break down. Lots of it in the subsoil of the small islands with the deep humus, not so much the big islands. So the deep humus on the small islands is because we have lots of this bound eugosterol and not so much of this free eugosterol. And finally, if we look at glucosamine, the active protein constituent of fungi, which makes up our chitin, lots of it in the subsoil of the small islands with the really deep humus. So multiple lines of evidence that this deep humus is due to fungal contributions rather than plant contributions. So we've been trying to, we could get further insights by looking at the types of fungi that lead to this organic matter formation. We've been doing this by looking at the fungal communities, by getting soil samples from our islands and sequencing them using PCR techniques such as the Illumina platform to understand the fungal communities that are present in the system. Basically, this is an ordination diagram. It's not as complex as it looks. For those who are not familiar with this type of approach, what this diagram shows is each of these symbols is a different soil sample. Points that are closer to each other have fungal communities that are more similar. Points that are further apart have fungal communities that are more different to each other. The big symbols are big islands, little symbols are little islands. The blue represents deeper down the soil profile. The green represents nearer the soil surface. So this is the big islands here. And these are fungal communities that are associated with high productivity, low carbon storage. These are small islands which have fungal communities that are associated with high carbon storage. So this diagram is the diagram that I had in the previous slide. These other three slides represent subsets of the fungal community plotted out in exactly the same ordination space. So this diagram shows a group of fungi we call the basidiomycetes. These are fungi that are associated with roots of trees. Lots of them on the big islands with a shallow humus, not so many on the small islands. These fungi produce residues that we know break down really fast. This is ascomycetes, which are other fungi that are associated with the roots of shrubs and produce residues that are well defended and break down really slowly. Lots of them on the small islands with the deep humus, not so much on the big islands. So the deep humus or the small islands is because of lots of these tough to decompose fungi and not so many of these. And what this shows here is fungi that are associated with dead leaves and break them down. These fungi are most abundant in the upper part of the soil profile, which according to our model I showed earlier, is the only part of the soil profile where the organic matter or humus is derived from dead leaves and not from dead fungi. So we've explored this theme. I'm not going to present any more data now, but we've done several other experimental studies which show that, in fact, different fungi, that the fungi community is a major contributor to organic matter formation in the system. We've done this, for example, through long-term removal experiments. We removed subsets of the flora to remove different types of fungi to understand what the fungi are doing. So based on these sorts of studies, this is now how we understand our system now works. This is what we find on big islands. We have fungi that form residues that break down really fast. So lots of CO2 gets released, lots of nitrogen gets released, and the plants grow really well. Not so much carbon and nitrogen stored in the soil. These are other sorts of fungi that produce residues that break down really slowly. Not much carbon and nitrogen release, so the plants don't grow very well and they're kind of stunted, and there's lots of carbon and nitrogen stored in the soil. This is relevant to global change issues because this is the system we're moving to in the boreal forests that are less inhabited, and where fire frequency is resulting as a result of climate warming. And this is what's happening in the more inhabited parts of the boreal forest, where fires are being intentionally suppressed and we've got a lower fire frequency. Okay, I'm going to move to a further longer time scales still by looking at soil gradients that are formed over geological timescales. I'll start by introducing work we've been looking at what we call chrono sequences. These are simply sequences of soil of different ages since they formed. So we have a disturbance which creates soils of different ages. We're looking at 6 of these systems around the world, ranging from boreal to temperate to subtropical. They're all long-term. They range in age between 6,000 and 4 million years. And across each of these sequences is relative constancy of all drivers except the disturbance that set the chrono sequence in motion. So I'll briefly walk you through them. The first one is the one in northern Sweden. It's the one I've been talking about for the last 15 minutes or so. It's a fire-driven system, sites of different ages since the last fire. One in Alaska, also in the boreal. This is a sequence formed by retreat of a glacier. So we've got a glacier that's retreating, leading to soils of different ages, from ones currently forming to ones that are 14,000 years old. 2 in New Zealand, one in the Franz Josef Glacier sequence, which is a popular tourist site. Maybe some of you have been there. There's a large retreating glacier, but it's also created a series of soils ranging from the current to those 140,000 years old. Another one from New Zealand, the Waitutu Marine Terrace System, surfaces of different ages coming up from the ocean, gradients of 500,000 years old. And 2 subtropical ones, one in Queensland, Australia, surfaces of different ages caused by wind movement of sand that spans 500,000 years, and one, a well-characterized one on the Hawaiian archipelago that spans 4 million years since volcanic eruption, since volcanic deposits that started the chrono sequence. So this represents the very earliest data we got from the study. What this shows is tree basal area, some crops sectional area of all the trees, versus time since the chrono sequence started. For Australia, Sweden, Alaska, Hawaii, and the 2 New Zealand ones. So for all 6 systems, there's a rather recurrent trend. Between 2,000 and 12,000 years since the chronosequence started, we have a reduction in plant biomass. The high biomass climax forest does not persist forever, it eventually degrades. And we have multiple lines of evidence, of which I just put one here, that show that this is tied up to the relative availability of nitrogen versus phosphorus. Here as before, the x-axis is time since the chronosequence started. Y-axis is the nitrogen to phosphorus ratio of the substrates or soil that the trees are growing in. The dotted line represents a type of Redfield ratio, the ratio above which phosphorus is limiting relative to nitrogen. So for each of these sequences, the points go above the dotted line at about 2 to 12,000 years. In other words, the time at which biomass is declining. So biomass declines as a result of phosphorus limitation rather than nitrogen. Now, we've reinforced this by various other studies, and the general trend is that across these chronosequences, nitrogen can be input because bacteria can convert atmospheric nitrogen gas by nitrogen fixation to forms that plants can use. Phosphorus cannot be biologically replenished. We start off with phosphorus in the rocks. Eventually, the phosphorus in the rocks gets used up. So phosphorus eventually must therefore become limiting relative to nitrogen. Now, this trend is also being picked up in a number of other chronosequences worldwide as well. This represents a global study that we and various others have contributed to, which involves chronosequences from a whole range of sites around the world. And there's a general trend that as chronosequence age increases, the substrate entropy ratio also increases because phosphorus generally always gets eventually more limiting than nitrogen. Now, most studies that have been carried out on this have generally looked at relatively low diversity systems and often in moist areas. So we're also interested in how this sort of pattern varies across climate. So this is something we've been exploring on across contrasted climates in Western Australia, together with NTU group, formerly based in UWA. So we have chronosequences of 2 million years age in each of 4 contrasted climates. Jurien Bay, that's really dry, Warren that is really wet, and these other 2 that are somewhere in the middle. So Jurien Bay, we go from young to old, and there's no trees because the system is too dry to support trees. Warren, which is really wet, we have really big trees, and the other 2 are somewhere in the middle. This shows just some of the data we got. We also got a whole bunch of below-ground data, but I won't present it here. The trends are generally the same. What this shows is that in the dry system, there's no real trend. When we go to the wet site at Warren, the plants are perhaps not surprisingly a lot bigger. There's more, the high leaf area index or plant biomass is higher, but we have this humpback relationship. Nitrogen limitation, then the nitrogen increases, biomass gets big, eventually phosphorus gets limiting, and biomass goes down. So what this suggests is that these patterns we find of phosphorus limitation occur more in wet areas than in dry areas. Dry areas, biomass is constrained by aridity, lack of moisture, and therefore nutrient limitation effects don't express themselves quite in the same way or quite as strongly. So strong climatic drivers of how these long-term chronosequences express themselves. So sticking with the climate theme, I'll now just present one final piece of work here. And this involves climate gradients. Now, if we're to understand climate warming effects, we have 2 main empirical approaches at our disposal. First of these is formal experiments. This involves this approach involves manipulating the climate somehow. For example, we could use open-top chambers, which we place on the ground in the tundra or above or in mountains, and this warms the ground up by about 2 to 3 degrees Celsius. We could use more controlled approaches like infrared lamps or heating cables in the ground to warm the system up. Or we could use more controlled approaches still, such as these mesocosm facilities, the ecotroid facility at the CNRS in Montpellier. Now, these studies are really helpful in understanding the physiological processes that govern ecosystem response to climate change and how they function. But they have one limitation, and that is that they are inherently short-term, whereas climate warming occurs over much longer timescales, decades or centuries. So the other approach we have is natural gradients, such as elevational gradients. And in this context, it's relevant that a 500-meter range in elevation represents the same temperature change as we'd expect through climate warming over the next century or so, about 3 degrees Celsius. Now, elevational gradients have been used for understanding global warming effects and in many studies on range expansion and population dynamics, but until recently, they've been used rather sparingly for understanding ecosystem responses. This represents 3 elevational gradients that have been used for this: Bisco in the Swedish tundra, Colorado Rockies, and one in the Peruvian Andes. The idea is you take measurements at different elevations and use the low elevation stands to predict what the higher elevation stands will become as a result of climate warming. So these sites, for example, turn more into these ones in the next 80 to 100 years. The work that my colleagues and myself have been carrying out in the past 15 years has been largely in the Swedish subarctic, in the Abisko region. Now, this is a region that's being disproportionately impacted by climate warming. We're expecting about a 5-degree temperature change over the next century, or 4 degrees by the end of this century. And all we have is plots of different elevations changing in temperature. So we use this site with the purple flowers to predict what this site will be like in about 80 years' time. This represents some of the first data we got from this work, led by my then PhD student, Maya Sundqvist. What this shows is nitrogen to phosphorus ratio in the foliage and dead leaves for 3 woody species: betula, birch, impenetrable crowberry, and vaccinium, and 2 herbs, Trollius and viola, going from high elevation to low elevation. As we warm from high to low, the entropy ratio moves in a fairly consistent direction, indicating that there's some predictability about how the relative balance of phosphorus and nitrogen is affected by warming. Now, we're interested in whether this was specific to this environment or whether this was a globally recurrent trend. So we then looked at other elevational gradients around the world as well, and some work that is headed up by my then postdoc, Jordan Mayer. We selected 7 sites around the world where we have strong elevational gradients across tree lines. The British Columbia and Colorado in the Rocky Mountain Range, Switzerland and Austria, Hokkaido in Japan, Nelson Lakes in New Zealand, Kosciuszko in Australia, and Chile and Patagonia. For each of these regions, we set up several replicated transects, and for each of them, we measured a bunch of properties above and below the tree line at different elevations. This informs whether the effects of warming are likely to be just due to the presence versus absence of trees, whether there's an effect of temperature independent of trees, and whether these effects are recurrent across different regions. This shows what we found with regard to plant foliar and root nitrogen at the community level. Each y-axis is standardized values of foliar nitrogen and root nitrogen, and each of these curves represents a different location. So as we warm things, we go from high elevation to low elevation. There's a predictable trend in nitrogen increasing. This is because as you warm things up, nitrogen gets released more by microbes from dead leaves and from the soil. So there's more nitrogen for the plants, and the plants suck up more nitrogen. We did not find this for phosphorus, and phosphorus we got a rather more complicated story. And when we considered nitrogen to phosphorus ratio, which was our focal variable of interest, we found varied responses. What this shows is foliar entropy, root entropy. For high elevation, low elevation. So here we get a convergence. As we cool things down, at different locations, all converge to a narrower range of values. To put it in another way, when you warm things up, they diverge. You get a stoichiometric divergence. And this is because at cold temperatures, the entropy ratio is controlled by temperature and certain physiological processes that constrain the entropy ratio to a narrow range. When you warm things up, these constraints are no longer important, and local factors become more important. Things like rainfall, geology, parent material, soil fertility, biogeography. This divergence was also statistically significant for root entropy. The pattern isn't as strong, but there is a wider divergence here than there is here. We were also interested in the mechanisms by which temperature affected in foliar nitrogen and phosphorus. And we explored this through structural equation modeling. For those not familiar with the approach, this is a type of path analysis where we look for the strongest relationship that explains a particular pattern. This is the model that we set up. So you considered elevation associated temperature, tree presence absence, soil fertility, microbial properties, nutrient availability, and soil plant nutrient status. This is the most parsimonious model that we ended up with, and therefore the model that we assumed was the correct one. There's a direct effect of elevation, associated temperature on plant nutrition, but there's also a series of indirect effects manifested through tree cover, organic matter, microbial biomass, and soil nutrients. So multiple pathways through which temperature affects these plant nutrition variables. So what we find across the 7 regions is that as we cool things down, there's a decline in plant nitrogen, but not phosphorus. And this leads to a convergence at colder temperatures of entropy ratios across regions. Warm things up, things diverge because local factors are more important. Also, there's various pathways through which these effects occur, not just directly through temperature, but also indirectly via soil nitrogen, organic matter, and the soil microbial community. And there's quite some consistency across contrasting temperature regions worldwide. Now, this is simply an observational study, and observational studies have their advantages. We can look at things and answer questions on geological timescales, but they have shortcomings because of confounding factors. The best of both worlds is to combine observation and experiment on the same study. And this is something we're currently carrying out through the WARM network, a large multi-country study with multiple PIs, coordinated by various people, including our colleagues, primarily our colleagues at the University of Michigan. At each location, we have different elevations, and at these elevations, we have overtop chambers set up to experimentally warm things up. These are mini glass houses that warm the ground up by about 2 degrees. So we have elevation, warming at each temperature, and also removal of dominant plant species at each temperature to look at the effect of plant species on these warming effects. We're just beginning to get data now and we're beginning to synthesize it. We don't have much of a story now, but hopefully if I give the seminar in 3 years' time, we'll have a more global story to consider with regard to these, how these manipulations play out. So just before I wind up, I'd just like to leave you with 3 take-home messages. The first of these is through the studies I presented, and there's a lot of studies out there that demonstrate this. If we're to understand the way that ecosystems function, we really need to consider both the above ground and below ground components and the feedbacks between them. Historically, there's been a tendency to consider either what happens in the plant community What happened in the soil in isolation? But over the past 15 years in particular, there's been an enormous recognition that to understand things, we need to consider how they feed back to one another. Second, this combined above-ground, below-ground approach enables tremendous insight about how global change phenomena may affect things. I've illustrated 3 global change examples, one involving invasive organisms, which are a major problem worldwide, one involving fire, which is again a major global threat in many parts of the world, particularly under climate warming and in a prolonged summer drought. And also climate warming, for example, through our climate gradient work. And thirdly, although we look at processes in the short term, these generate insights into much longer processes, including those that operate over thousands of millions of years, including geologic timescales. Beginning with the figure I started, we look at processes in the short-term frame, physiological processes, decomposition, vegetation change, but these both respond to and drive processes that occur over thousands and millions of years. And finally, just in finishing up, I'd just like to thank the various colleagues who have contributed to the ideas and data given in this presentation. Thank you.
Thank you, Prof. David, for such an insightful talk. Now I would like to invite our co-host Ruchi from the School of Biological Sciences to host the Q&A session.
Thank you, Shivam. Let's move on to the next segment of our seminar, which is Q&A. So attendees who would like to ask questions to Professor can ask their questions using the raise hand feature at the bottom of the Zoom screen. And when your name is called, please unmute yourself to ask the question. Alternatively, you can also send your questions in the Q&A tab. So while the audience is coming up with their questions, we would like to ask some questions to Professor Wardle.
Please.
Yeah, so the first question was sent by a participant, Lynette Leong, at the time of registration.
Yeah.
And the question is, how accurately could you use the data you have to project the effects of climate change on these environments? And could this be one of the key evidence to evoke more efforts on an international or governmental level to help with the climate change?
Okay, I think in understanding the effects of climate change in ecosystem, there's many pathways of the truth. And what I put up here is just one of many pathways. Our pathway involves using long-term gradients to infer what climate change will actually do. And this can inform, for example, in terms of mechanisms that occur in the long term. We know from our work and the work of some others that if you warm things up over decadal to century timescales, you consistently get greater mineralization of nitrogen, greater nitrogen nutrition of plants, changes in nutrient balances, and feedbacks involving plant growth. Going to the fire example, we know that fire is a major threat in many systems worldwide. It's becoming more prevalent as we get warmer conditions and more summer droughts. It's not something we can hide from. I mean, we see it in our news bulletins as various parts of the world undergo through their fire seasons, and you get tremendous fires in parts of North America, Australia, Mediterranean Europe, Northern Europe, Siberia, Alaska, and such like. Our results, along with many others, inform on what happened to the carbon balance of the system and also the mechanisms underpinning it. So I think we are at the stage that some of these data sets with judicious analysis can inform predictions. We contributed to a meta-analysis that came last year that used climatic fire gradients around the world to understand what effects fire suppression and fire frequency has on carbon storage. There is predictability, and I think it can inform policy. We can't answer everything yet, but I think we're making enormous steps.
Thank you, Professor. There's another question. So what would be some advice that you would like to give to a PhD student who has just started his or her research journey?
In this topic, I think the main advice is find something interesting and find something you enjoy. If you're not enjoying it, you won't have much fun doing it, and it will not go as well as if you are enjoying it. But also find something interesting, find a question that hopefully opens a new part of the box, looks at things a different way, and go for it.
Thank you, Professor.
Yeah.
So here we have a question from our live audience. So the question is, do you think that the below ground fungal communities are plant host specific, or are they controlled by soil type nutrient content?
Both. Yeah, both are important. They can look at two pools of fungi, because they're the two that are essentially, I mean, it's very simplistic, but essentially you've got fungi that are associated with plant roots, and these include pathogens, and they include mycorrhizal fungi. The fungi that most land plants have that have mutualisms with which bring them nutrients. These are controlled to some extent by the host, but not entirely, and there's different sorts of plants that have different sorts of fungi associated with them. So there is some specificity. But it's also controlled by soils, particularly for saprophytes. I mean, some fungi are adapted to soils which are highly nutritious, low nutritious, have lots of clay, not clay. This is what we're looking at now is peat swamp forests. We're interested in, for example, soil moisture in these forests and how that regulates the fungal community. But some mycorrhizal fungi like being very wet, some don't. So there's multiple drivers. So both are important. Interestingly, we're still struggling. I mean, there's some really good work coming out, but we're still struggling to look at the relative importance of host-driven versus soil-driven factors. But they're both important. And there's also an indirect pathway as well, because soils also influence the types of plants we have, and that also influences the fungi.
So the next question is, how effective would it be to investigate the geochemical cycles occurring in urban forests and parks to understand potential effects of climate change in vegetation at larger scales?
You look at local scales. It's relative. I mean, it's certainly doable. I mean, we're involved in work actually which has been carrying out in Singapore that's been headed up by our colleagues in Finland, where we've been looking at 3 cities in depth. We're looking at Helsinki, which is a boreal city, Baltimore, which is temperate, and Singapore, which is tropical. We've been looking at various processes in forested and non-forested parks in each of the 3 cities. And you could answer this paper that's we've just had come out in Global Change Biology. That's was of course the first results from this for those who are interested. But basically at that level, it's reasonably straightforward. We can get soils, we can sequence them to look at the fungal community, we can look at the soil properties, link them together, look at different climates and see how they respond. In terms of scaling up to bigger questions, and I think we still have a way to go. We have 3 cities. It'd be nice to have 30. I think when you've got more, you could answer more general questions. But I think it's doable.
There's another question. Given your research showing old growth on small islands sequesters more carbon over long time scales, is there a way of transplanting those fungal communities to younger forests to aid in carbon sequestration?
Then probably not. I think they die. And the reason is that if you put them to a more productive system, you've got fungi that are more productive, and they would kill out or compete out the less competitive fungi. So one of the sort of more general trends in ecology is productive systems tend to have organisms that are more competitive than unproductive systems. So if you're an unproductive system, you've got to focus on getting resources and being really well defended, being very persistent, but you won't be so competitive. You go to a productive system, you're investing in rapid growth and you compete things out. That's why, for example, if you put a fertilizer on a lawn, you get fewer species. Your most competitive species compete everybody else out. You take an unproductive plants, say microbes from an unproductive system, put them in a productive system, the fungi are more competitive on the productive system, they'll compete out the unproductive fungi and kill them. It must be able to do that, but I think you'd have to go further than that. You'd have to suppress the competitors.
So there's one more question. Could you give more details on the impact of C to N ratio on the soil microbial communities, and what kind of species get enriched under lower C/N ratio?
Okay, I mean, well, there's really, really three parts. There's carbon to nitrogen, carbon to phosphorus, nitrogen to phosphorus. So they also have they all have their interesting stories. But if we focus on specifically on carbon to nitrogen ratio, then generally plants with a high carbon to nitrogen ratio and do not so produced by plants, maybe in more stressed environments, plants that grow slowly, plants that have less nutrients in them. They produce residues that break down slowly, and they support microbes that are tolerant of nutrient poor conditions. Often microbes that are turn over less, maybe eaten less, cycle nutrients less, grow more slowly, and hold on to resources rather than cycle them. When you've got more nitrogen present, and you find this, for example, if you add nitrogen fertilizer to an ecosystem, you've got microbes that are adapted for, well, adapted for faster decomposition of more labile substrates. They may not decompose tough substrates as well, but they decompose simple substrates more easily, and you get faster nutrient cycling going on. It does get more complicated because having more nitrogen also impairs certain enzymes that break down lignin. So when you've got more nitrogen, labile substrates break down fast, others break down more slowly, based on the types of fungi they select for.
Thanks, Professor. So there's another question. How applicable are these findings to tropical ecosystems where species density tend to be higher? Will there be various effects exerted by different neighboring plants in high biodiversity system?
That's a very good question, actually a very tough one that we're still trying to get our head around, because high diversity systems are inherently more difficult to study simply because there's more species to work with. The conventional wisdom is, yes, things do compete in the tropics. Do they compete as strongly as they do in the temperate? I think that's an open question. It's rather more difficult to manipulate, to do manipulative experiments, but there is some observational studies that indicate that different plants, groups of plants exclude each other out, maybe at the functional group level rather than the species level. And I mean, there's long-term data, for example, from the Forest Geo Network. I mean, there's been work at Barrow, Colorado Island that's been using some really detailed work looking at neighborhood effects and how that affects competition. So yes, things do compete. They compete more or less in the tropics. I don't know.
So I think we can take one more last question. So this question is, how useful do you think are microbial community models to study long-term patterns and drivers of ecosystem functioning, but in short time scales? For example, most relevant bacteria in activated sludge or wastewater treatment have generation time of less than 24 hours. Hence, a few months on this system would represent around tens or hundreds of generations of many different microbial taxa, which in terms of generations could be equivalent to hundreds of years of plants in a forest.
Yeah, well, I think of timescale is really interesting. That I mean, what we consider a long-term experiment for trees would be thousands of years because you need multiple generations. You get the same result with bacteria in, say, a few days. I think in terms of understanding what's happening at the local scale, yes, those sort of models are really useful. At longer time scale, there's many other processes that feed back to it. So, I mean, basically the microbes might be sort of helping stimulate or impact these longer term processes. But at the same time, these longer term processes feed back and impact upon the microflora as well. So I think if you're trying to project things over thousands of years, then you're looking at having to introduce other models that incorporate these longer term processes. So it depends on the question you're asking. But like a lot of things, models are one of many ways to the truth.
Okay. Thank you, Professor. So thank you for delivering such a very insightful and thought-provoking talk. Also, thank you for patiently answering all our questions.
Well, thank you. So it's been a pleasure.
Thank you. I'd also like to thank and congratulate all the organizing teams for this wonderful event. And we would like to also like to thank the audience for attentively and actively participating in our talk and making this a very interactive session. So I would like to request the participants to please scan the QR code, which will shortly appear on your screen, and provide their valuable feedback. You may indicate the need for endorsement in that attendance form itself. So this brings us to the end of the seminar. Thank you all. Thank you all for attending the seminar, and take care and stay safe.